Hello everyone. Today I'm going to discuss with you about the metabolic pathways. We have discussed some of the metabolic pathways during our previous classes. As you all know that these pathways come together like puzzle pieces at certain points due to some intermediary compounds. It all starts with the meal. As we consume our meal, it is digested by our stomach and then the nutrients are absorbed from the intestine in the form of proteins, carbohydrates, and fats. And from the intestine, these nutrients are absorbed into our bloodstream that goes to liver as a first thing, and then from there it distributed to rest of the body cells. And every cell in the body gets the nutrients according to their need and demand. Today, I will be focusing on the glucose metabolism with respect to our midterm demand. So, from the bloodstream, the glucose will enter into the different cells. So, I'm not specifying at this point that which cell it's gonna go and how the cells are different. For example, it's a general cell. Maybe it could be a liver cell, or it could be a muscle cell, or any other cell that a cell in the body that utilizes glucose for energy production. Once glucose enters into the cytoplasm of the cell, it has pretty much defined fate. Either it will be converted into glycogen if it's a liver cell or muscle cell, or if it's a regular cell which just utilizes glucose, it will be utilized to produce ATPs via glycolysis. Here's a recap of glycolysis. So glucose will be converted at the expense of one ATP molecule to glucose 6-phosphate, and from there it will be converted into fructose 6-phosphate. I didn't mention the enzymes because it will be too much for me to record everything and even to mention all the enzymes. But we all have discussed those things too many times, and we all have all the slides available where we can just go back and look for the enzymes for each step. I'm just giving a kind of overview of some of the important things over here. So fructose 6-phosphate will be converted into fructose 1,6-bisphosphate. From there it's a 6-carbon compound. From there it will be dissected into 2 3-carbon compounds, like could be glyceraldehyde 3-phosphate or dihydroacetone phosphate. And from there an NAD will be converted into NADH, and with the production of NADH, the glyceraldehyde 3-phosphate will be converted into 1,3-bisphosphoglycerate, and from there another ATP generation, like the first ATP generation will happen, and the 1,3-bisphosphoglycerate will be converted into 3-phosphoglycerate. So remember, these are 3-carbon compounds, so they are in two times, right? So 2 ATP will be generated here, and then from 3-phosphoglycerate it will be converted into 2-phosphoglycerate, and from there it will be converted into phosphoenol pyruvate, which is also commonly known as PEP. So PEP has importance as which I will discuss later, that how the in the gluconeogenesis, how the pyruvate takes a shunt through the mitochondria and get back to PEP. And once the PEP is then converted into pyruvate, and there will be an ATP generation over here. So glycolysis has 2 other intermediary compounds that can enter into this pathway. For example, one is the lactate. So lactate can enter into this glycolysis and then TCA cycle. At this point, it will be converted into the pyruvate and there will be an NADH production. Other than that is the glycerols. So glycerol can be converted into the glycerol 3-phosphate by the expense of ATP, and then the glycerol 3-phosphate will be converted into dihydroacetone phosphate, and then one NADH molecule will be produced over there. So at the end of glycolysis, pyruvate will enter into the mitochondria, that is also called powerhouse of cell. So from here, once pyruvate is entered into the mitochondria, it will be converted into the oxaloacetate, and this will also be on the expense of ATP. And from oxaloacetate, it will be converted into citrate, and from citrate it will be converted into alpha ketoglutarate and there will be a production of NADH, and from alpha ketoglutarate it will be converted into succinyl CoA and another NADH molecule will be produced over here, and from succinyl CoA to succinate an ATP molecule will be produced, and from succinate to fumarate an FADH2 molecule will be produced, and fumarate to malate. And malate is important over here because it can shunt out of the mitochondria and it can go back to the PEP to start the cascade of the gluconeogenesis, which I will discuss shortly. So from malate it will go back, the circle will go back to oxaloacetate and another NADH molecule will be produced. So other than the carbohydrate which are being processed over here through pyruvate, all other amino acids and lactate and propionate, which is a volatile fatty acid, those are those are so other than the carbohydrates that are included into the TCA cycle via pyruvate, other amino acids, almost all amino acids, and propionate as a volatile fatty acid and lactate is also being processed through this Krebs cycle or TCA cycle. And I have mentioned over here the name of the amino acids that through some reactions are converted into different components of the TCA cycle. So that's why it's so important. And this is, I guess, is one of the reasons the mitochondria is called as powerhouse of cell. And the other thing is obviously is the electron transport chain from where ATP is produced by utilizing the NADH and FADH2. But these are also important where different amino acids and volatile fatty acids, propionate specifically, it's very important in ruminants digestion to produce energy. And after that, if we follow this malate, which is kind of being shunted out of the mitochondria, once out of the mitochondria, malate will be converted into oxaloacetate and another NADH molecule will be produced over here, and then oxaloacetate will be converted into phosphoenol pyruvate or PEP. And the enzyme over here is an important one, so the name of the enzyme is PEP carboxykinase. And in this step, a GTP molecule will be consumed and carbon dioxide will be released. And from PEP, the gluconeogenesis will start, and it's basically almost opposite to the glycolysis, but the enzyme, those are different. Are one is over here that the malate shunt has to be taken by the pyruvate to convert it back into the PEP. So that means the pyruvate kinase wasn't able to convert pyruvate directly back into the PEP. So that is kind of one of the irreversible step over here, and then from PEP the cascade will start going back towards the glucose, and PEP will be converted into 2-phosphoglycerate, and then from there 3-phosphoglycerate, and 3-phosphoglycerate will be converted into 1,3-bisphosphoglycerate by the expense of an ADP molecule, and from there 1,3-bisphosphoglycerate will be converted back into the glyceraldehyde 3-phosphate, and over here one NADH molecule will be expensed. And this is the point where, for example, if there is a glycerol available in the cytoplasm, it will be converted into glycerol 3-phosphate, and then from there DHAP, and DHAP with the combination of glyceraldehyde can be converted back into the fructose 1,6-bisphosphate. So fructose 1,6-bisphosphate will be converted into fructose 6-phosphate, but the enzyme here is fructose 1,6-bisphosphatase. It's not the same enzyme which was phosphofructokinase when fructose 6-phosphate was converted into the fructose 1,6-phosphate during glycolysis. And fructose 6-phosphate will be converted into glucose 6-phosphate, and glucose 6-phosphate will be converted into the glucose, and there is a specific enzyme named as glucose 6-phosphatase that will catalyze this reaction, and the glucokinase or hexokinase are not able to convert the GCP and G6P back into the glucose. So both these steps are also irreversible steps of the glycolysis, as going back during the gluconeogenesis, different kind of enzymes are required for both of these steps. So during gluconeogenesis, once glucose 6-phosphate is produced, it can be converted into glucose 1-phosphate, and from there it can be converted into UDP glucose, and then UDP glucose will be converted back to glycogen by using glycogen synthase, and these glycogen synthase is activated by the presence of insulin in the liver and muscle cells, and it is deregulated by the presence of glucagon. The NADH and FADH are energy analogs that are produced during the Krebs cycle and glycolysis. They will be then utilized through the electron transport chain to produce ATP. So over here, if we start looking into the electron transport chain, the complex 1 NADH will be converted into NADNH, and hydrogen will be expelled out of the mitochondrial matrix into the intermembrane space, and from there complex 2 FADH2 will be utilized and converted into FAD, and then from in the next complex 3, one hydrogen will be pumped out of the mitochondrial matrix into this intermembranous space. And same with the complex 4, one hydrogen is pumped out, and within the mitochondrial matrix, 2 hydrogen ions will combine with an oxygen atom to produce water, and then at the last electron transport chain is ATP synthase. So that's a major enzyme that gives the mitochondria name of a powerhouse. So these hydrogen ions which were pumped out into these intramembranous space of the mitochondria will then pump back inside the mitochondrial matrix through the ATP synthase, and then an ADP molecule will be phosphorylated into an ATP molecule. So that's how these reducing NADH and FADH2 will be utilized to produce ATP in the mitochondria of cells. Simultaneously, in the cytoplasm of the cell, there is an other important pathway that is happening and is called as pentose phosphate pathway. In this pathway, the glucose 6-phosphate is converted into gluconolactone and NADPH is produced, which is another important energy analog, and then gluconolactone will be converted into 6-phosphate gluconate, and that will be converted into ribulose 5-phosphate. So ribulose 5-phosphate is, if catalyzed by the ribulose isomerase, it will be converted into the ribose 5-phosphate, and if it is catalyzed by the ribulose epimerase, it will be converted into the xylose 5-phosphate. So from going from 6-phosphogluconate to ribulose 5-phosphate, we lose one carbon in the form of carbon dioxide, which I forget to mention over here. So now the 6 carbon molecules are converted into 5 carbon molecules over here. And then this ribose 5-phosphate and xylose 5-phosphate in the cytoplasm of the cell can combine together to form a GA3P glyceraldehyde 3-phosphate or pseudoheptulose 7-phosphate, and then the pseudoheptulose 7-phosphate and GA3P again combine together to form a erythrose, which is a 4-carbon compound, and fructose 6-phosphate, which is a 6-carbon compound. And then this erythrose 4-phosphate can be combined or react with the xylose 5-phosphate to produce fructose 6-phosphate and glyceraldehyde 3-phosphate. So GA3P and F6P are the intermediary compounds over here, if you recall the glycolysis or gluconeogenesis. So that pathway has both of these compounds, which, like, these compounds can be utilized either to go back towards the glucose or towards the pyruvate as a result of glycolysis to produce more energy. This pathway is also important for utilizing the carbohydrates that are not 6-carbon compounds. As we eat a lot of different things that has different kind of polysaccharides, so it's not necessary that everything will be break down by our stomach into the 6-carbon carbohydrates. So some of the 5-carbon, 4-carbon, 3-carbon carbohydrates are also produced and absorbed into our bloodstream. So this is the pathway that makes our body cells able to utilize those carbohydrates and to shunt them back into the glycolysis uh, cascade to produce ATP for our daily use. So I'm gonna give you a recap of how the blood glucose is regulated. So as we all know that there are two major hormones, one is insulin and other one is glucagon, that are responsible for maintaining the blood glucose level in a certain range. So our blood glucose is maintained in a certain range unless we eat a meal, and then after taking a meal our blood glucose increases to a certain level, and that stimulates the pancreas to release insulin into the blood. And once insulin is released into the blood, it brings the glucose down by converting the glucose into glycogen via liver cells. And then if we go to sleep for a longer time, like during night for 8-10 hours, so our blood glucose level drops as we are not intaking any kind of food or meal during sleep. So once the blood glucose level drop to a certain limit, then the glucagon is produced from the pancreas that will result into the production of glucose from glycogen reserves into the muscles and liver, and that will result in increased blood glucose level. So that's how our blood glucose level is maintained throughout the time. And it's not like after meal insulin will be released and after sleep glucagon will be released. They are released at a basal level at every time side by side, but when we take a meal, obviously there is a peak increase in glucose, so that needs a peak increase in insulin release. And same as with the starvation or sleep, there is quite a lot decrease in the blood glucose level, so there is a peak glucagon production is required to combat that and to produce the glucose from the glycogen reserves in our liver and muscles. Thank you all for your patience and listening to me. I'm not sure, this is my first time ever recording on this PPT version. I'm not sure if I will be able to put all these things together in a fine way, but I hope I tried to explain that how different pathways are interlinked through different intermediary compound, and how the basic purpose of human cell is the production of ATP or the production of energy. So how everything is, or every pathway is serving towards that purpose, and ultimately our cells are able to produce ATP to maintain our body for our reproduction, for daily routine and everything. Thank you once again.